Hi everyone, in this video we're going to continue exploring different types of nucleic acids, specifically the nitrogenous bases that are involved in a single nucleotide structure. Now, nitrogenous bases typically fall into two different structural categories, and this is purine and pyrimidine. And this is important because it really relates to how the nucleic acids are able to maintain a consistent shape and width, especially when they are involved in a double helix sort of structure in a DNA. Firstly, we have pyrimidines, and they're basically single carbon ring bases. And different types of pyrimidine bases include cytosine, thymine, and uracil. Secondly, we have purines, which are double ring bases, and this is basically adenine and guanine in the context of nucleic acids. Now, the very important rule that we have to remember here is that purines and pyrimidines bind together and they pair together. So it can't be purine and purine, it can't be pyrimidine and pyrimidine. It has to be a purine and complementary pair being pyrimidine. And the reason for this sort of rule is because we need to maintain the sort of structural consistency as we form a double helix in a DNA structure. If we have this sort of larger base pair, it really has to kind of go with a smaller base, which is going to be pyrimidine in the case. So we have a larger purine and a pyrimidine kind of to maintain this sort of uniform width within the helix. And this is essential for maintaining a stable nucleic acid structure, especially in the context of DNA. Right, so this is basically a more organized overview of which bases are present in DNA or either RNA. This is something that we probably do know in DNA. We have A, T, C, G pairs, and in terms of the one-ring bases for the pyrimidines, we have C and T. Purine, we have A and G, basically the same for RNA except for the fact that thymine has been substituted by uracil here. One thing that I like to do when I'm remembering these sort of names is, especially in terms of having to remember the number of rings that are for pyrimidine and purines, I tend to just associate the one that has the longer length and name with the shorter length and base. So we have pyrimidines, and it's a one ring, it's short. We have purines, a short name, but it's a long sort of base, or a larger structure, a two-ring base. Right, in this in this slide, we're specifically going to look into the detailed properties of pyrimidines, which are single carbon ring nitrogenous bases. Now, pyrimidines, as I've spoken of, they're one ring nitrogenous bases, and they contain cytosine, thymine, and uracil. Now, cytosine specifically is found in both DNA and RNA, which kind of makes it a very common base for both of the nucleic acids. Thymine specifically is found only in DNA, and it pairs with an adenine base within DNA. And uracil is also found in RNA specifically, and it's kind of substituting thymine in that it pairs with adenine also. Next, we're going to look into purines. Now, purines are two-ring bases, and they can either be adenine or guanine. And there are two things that really matter here. First, structurally, purines are larger because they consist of two fused rings, whilst pyrimidines only have one ring, and that is why they must pair with another one smaller ring, pyrimidine, for the helix to have to maintain consistent spacing and width. And second, functionally, both of the purines, adenine or guanine, they are present in both of the DNA and RNA, so you can't necessarily use them to distinguish which is a DNA molecule or a RNA molecule. Right. Next, we have complementary base pairing rules and just bonding details in general. Now, complementary base pairing is the reason as to why nucleic acids can be copied so accurately and maintain such a stable structure. Now, the general rules for complementary base pairing is that an adenine must always pair with a thymine in a DNA, and an adenine pairs with the uracil in RNA, and guanine essentially pairs with cytosine in both of the DNA and the RNA. Now, these base pairings are held together by things called hydrogen bonds, which are just really sort of weak intermolecular forces, and it's important that not all bases pairs have the same number, and that if we were to have a cytosine and guanine sort of pair, CG pair, they would be linked by 3 hydrogen bonds, as opposed to when we have an AT pair or an adenine and thymine pair in the DNA, it's linked by 2 hydrogen bonds. Now, the difference in the number of hydrogen bonds really matters in terms of stability. CG pairs tend to be just generally much more stronger because of that extra hydrogen bond, and this is the reason why regions that are so rich of CG bases tend to be much more thermally stable. Next, we're going to move on to the structure of nucleotides. Now, as I've spoken of earlier, DNA and RNA, they're both polymers, and the repeating monomer is a nucleotide. A nucleotide is essentially built from 3 parts. We firstly have the phosphate group, we second have the sugar, a deoxyribose in DNA and a ribose in RNA, the difference being that a ribose has a hydroxyl group, whilst a deoxyribose in DNA has an absent oxygen, so only in hydrogen group, and a nitrogenous base, and this is C, G, and T in DNA, and the T is replaced by U in RNA. Now, it's very important to kind of keep the terminology really correct and precise here. A nucleic acid is made out of many nucleotides. A nucleotide is not equivalent to a base. It is a base plus a sugar and a phosphate. Now, this distinction is really, really important because we tend to kind of confuse these things, especially in an exam context. And it's really important because specific terminologies tend to give you hints about what the question is about and the scope of the question. So really do make that distinction. Alright, that is pretty much it for today. Next video, we're going to look into specific structures of sugars that are involved in the respective nucleic acids, as well as looking into the structure of nucleotides, which directly link to each of the helix structures and the structure of a DNA.